This lecture is going to serve as the intro to sampling strategies and sampling plans. We're going to go into a lot more detail on many of these items throughout this course, but this is really our chance to start thinking about how we plan sampling and what's important to think about. So when we think about water quality and what we're targeting, we're looking at the physical, the chemical, and the biological constituents. So that could be things, physical things like the flow, the grain sizes in the substrate. We might be looking at clarity or color of the water. For chemical, that's going to be concentrations of different contaminants or different constituents in the water. Whereas biological could either be bacteria and pathogens, or it may be looking at those biological and ecological communities. And we'll talk about all those things more throughout this class. So when we're setting up a study design, we have to take into account a lot of things, but really our key points that we need to think about are summarized in the process of developing data quality objectives. You'll listen to a more detailed talk on data quality objectives this week, but we'll touch on them here because it's such an important and integral part of planning sampling. So our DQOs, our data quality objectives, essentially state our study objectives. So, really what's the purpose? What are the appropriate types of data to solve a problem or make a decision that we need to make with that data? What's our tolerable error? That's really, really important for planning sampling. You have to balance the allowable error for a study with the resources, with the money and the time and the effort that it's going to take to complete that work. We can always say we'd like more data, but ultimately that comes at a cost. And then what's the quantity and quality of data that we need to answer the question at hand? So that's going to vary based on what questions we're asking and really what we need to know. So that DQO process is really going to guide us through, when to collect samples, where to collect samples. You'll see this term several times, but what's the tolerable level of decision error for our study? And essentially what that means is how bad is it going to be if we're wrong? So if we're making a decision based on the data we're collecting, how bad is it that we're wrong? So if we're trying to answer a question about the safety of drinking water or whether or not we need to close a recreational beach to swimming due to E. coli levels, those we have to have, very low decision error. We really need to be right because those affect human health. There are some there's some studies where maybe we're just determining background levels, or we're really just investigating a system where people's health or the health of an ecosystem aren't necessarily reliant on that data. And so maybe we can tolerate more error in that because we're trying to answer different questions. And essentially what that then gets to is a system of thinking through how many samples to collect and how do we balance that risk of being wrong with the cost. So when you think through these DQO steps and how we actually get there, we start out by stating a problem. So that's really important because it guides everything that we do. Even though it sounds very basic, we need to make sure that we have a clear problem statement. And from that, we'll then stem what's the decision that we make. So do we continue supplying that drinking water? Do we keep the beach open or do we close it? Do we build a treatment system or do we not? Those sort of questions are the decisions. And so then what are the inputs? What's the data that needs to go into those decisions to be able to make good decisions? We need to define study boundaries, and this needs to be both in terms of space, so in terms of, really like the geographic boundaries, but also time. So how long is our study going to go for? how much data do we need in terms of that time. We need to identify something called the decision rule, which is essentially how are we going to make the decision? At what concentration of E. coli do you shut a beach? How widespread does that have to be to shut that beach, for example? And then we need to set limits on decision errors. And that's essentially what I was talking about earlier, where, if it's a human health-based decision, then you can have very low decision error. Whereas if you are talking about, something with lower stakes, you may be able to have a higher decision error. And then we optimize the design to obtain the data that we need to make the decision with an appropriate level of decision error. So as part of this, we need to figure out where to sample. And so our sampling point has to be selected based on the information that we need to obtain. And so that goes back to that problem statement and the question and the data that we need to actually make a decision. So if we go back to this idea of a drinking water system, for example, we're going to want sampling points throughout our distribution system, throughout our water distribution system, to understand the variation in water quality through that distribution system. And we're going to want to make sure that we determine where we expect to have the worst water quality. So whether that be the areas with the oldest pipes, or that may be the area that's the furthest from the treatment system. There may be some specific knowledge about the system that lets you understand what the critical locations are, and so we'd want to include those in that study. Equally, in this example about whether or not to close a beach due to E. coli levels, we're going to want to have kind of a sampling strategy where not only are we sampling, across the geographic area of that beach, we also want to understand where are inputs that could be leading to high E. coli counts. And we're going to want to, direct our sampling towards, potential sources, and, with an understanding of currents of water, for example. We also need to think about when to sample. So again, like I said, we could design a study where we're taking, daily or weekly or monthly data, but that may not be necessary, right? That may be too much sampling, and often would be too much sampling depending on what question we're going to answer. So what we often do is think about this idea of sampling across a water year, or representing different stages. So looking at base flow versus high flow versus low flow. We understand that in most systems, the hydrology is a major driver of water quality. And so understanding the water quality at these different flow regimes is going to allow us to understand the variability in water quality across that water year. This connects closely to seasonality because, in Ohio, in a lot of temperate areas, that stage is going to vary based on the seasons. That said, there are other things that we might think about for seasonality. For example, we would expect to see the biggest algal blooms in, hot, low precipitation time periods in, July and August, maybe even into September, when you've had a big enough buildup of nutrients over the summer, you've got warm water, you've got very little flow through in lakes, for example, and that you're going to have, conditions then, in that time period that may drive worse algal blooms. And so you're going to want to focus your sampling, towards that part of the season, for example. We also want to think about current rainfall conditions. We typically aim to not sample when it's raining because we expect things to be transient. We expect things to be changing and dynamic during rainfall. But there are some systems where we also understand that, the first part of a storm is the most critical water quality condition. And so even though it's very difficult sampling, we'll sometimes do stormwater sampling to try to target that specific flow, specific flow and rainfall regime. We also need to think about then frequency of those samples to be collected at each site. So is it quarterly? Are we aiming for just a high flow, low flow sample this year, or are we aiming for more samples than that? Do you want it to be monthly, or do you want it to be based on, when it last rained, for example? So thinking through that frequency. And then for how long? Is a year's worth of data enough, or do you need multiple years, or do you not even need a year? Right, so as we actually then get down to picking sites, we have to think about and be very intentional about our site description and our site naming. So our site description is really, really important in our field notes. And it's really useful for return sampling events. So if we're monitoring changes at a site through time, we have to have very clear instructions, very clear landmarks to hit that same spot repeatedly. It it is much, much more difficult than it sounds to have a new crew who doesn't know sites follow somebody else's instructions to park, to access a site, and to sample at the right location, even with a GPS. So oftentimes we're really heavily reliant on GPS and GPS coordinates, but ultimately we need to have very clear narrative descriptions that allow a new crew to go in and access and sample at the same location. So we need clear site information. So that's things like exactly where you park, if there's an intersection nearby, using that as a landmark rather than something that might be more transient, like a broken down car or something like that, or the barking dogs. And we want to record our access information, so that includes permission to be there. And we have to have permission to be at all of our sites, and also directions for accessing the site. And then we need landmarks for where we sample. And so some of the really important things and some of the things that are difficult in the field are things like, are you sampling upstream or downstream of this tributary? It's pretty critical whether to get that right, to be in the right spot. But if somebody's forgotten to say, oh, you're sampling upstream of this tiny little side tributary, you can easily get that wrong, especially within the error of a GPS unit. So we want site information, we want a general description of that site as well. Site numbering is also a really important part of gathering data at a site. Site numbering is essential to allow individual site locations to have unique identities and to reference all of our samples back to those geographic locations. There are lots of ways of doing this. There's a separate video that you'll watch on Ohio EPA's River Mile system. What we'll often use is a modified version of this Ohio EPA River Mile system, where we abbreviate the sub-watershed names. We know that all the sites with that abbreviation are in that sub-watershed. So for example, Hewitt Fork would be HF, and then we would have a site number behind HF. And that site number will loosely correspond to the number of river miles upstream from the mouth of Hewitt Fork that site is. So for example, HF 010 is 1 mile from the mouth, whereas HF 045 is 4.5 miles up from the mouth. And so it's a slightly modified version of a river mile system that's pretty intuitive. So then when we're actually in the field sampling, we have to make sure that our field measurements represent the natural condition of the water system at the time of sampling. And so really what we want to do is we want to think through what we're doing to make sure that our field measurements and our lab measurements are representative of what was happening in the field at that moment. So some of the things that we do in order to ensure this, some of our quality assurance and quality control measures include calibrating instruments, testing those instruments, letting our instruments stabilize in the field, cleaning our equipment between sites, and repeating samples for accuracy. So those repeat samples are typically duplicate samples where we take two sets of samples with the same site ID, but different lab sample numbers at the same site, and we can compare the analytical data from those two samples to quantify our range of error for those samples. There are standard stabilization criteria for different meters. The values here are pretty standard across multiple meters. And essentially what the stabilization criteria do is tell you when you're at a number you can write down. When are you at a defensible number? Because all of the probes that we use for water quality sampling are essentially electrodes that that correlate an electrical signal to a concentration or a pH or a conductivity based on our calibration. And so these stabilization criteria essentially say, that electrical signal might bounce around a little bit, and these are the ranges of error that are acceptable. So then when we go into actually taking a sample, there are multiple different methods that we might use and some considerations we need to have for doing that. So most often when we're taking a sample, we are taking a grab sample. And so for a grab sample, we're going to take a sample in what we call the thalweg, which is the main flow of a stream at our sample site. And all of our sample bottles will typically be filled from one bucket dip in that main flow of the stream. We want to make sure we're not in a pool, that we're not in a riffle, that we're not in a backwater, that we're in a flowing area without an awful lot of turbulence. There are two other kind of key ways of taking samples. So an integrated sample is integrated over time. So this would be something like setting out an auto sampler and having it take a sample every hour for 24 hours, and all of those 24 samples are then mixed together, and essentially that would then represent the condition for that 24-hour period. A composite sample is similar, but instead of over time, it's over space. So we often do this with sediments, where you would take multiple sediment samples in a reach and mix them all together, and then take your sample from that mixture. You could also do that with water and composite, across a creek or along a reach. We also need to make the decision about whether we're going to filter our samples. Typically, most labs, the filtered and the unfiltered sample go in as two separate sample IDs, which essentially means it increases your cost or can double your cost. It's important to filter under turbid conditions, particularly to protect your equipment. You may need to filter in metal-rich waters where you have a lot of precipitated metal suspended in the water. And then scientifically, we often are filtering in order to distinguish between the dissolved and the suspended portion of often metals, but it can be of almost any constituent. We typically will filter using a 0.45 micron filter. That's pretty standard. There is some newer research where people use 0.2 micron filters. Those are operationally very, very hard to use in the field, but the research is showing that there are some colloidal fragments that are technically suspended and not dissolved that are between 0.2 and 0.45 microns. So depending on the study purposes, you may want to use a smaller filter. We then will also preserve our samples. So some samples are preserved with acid. That's common for metal samples, for nutrient samples, and sometimes for samples where we're going to analyze for something like ferrous iron. So we'll use a different acid depending on what the target constituents are. So typically for metal samples, we'll use nitric acid. For nutrient samples, we'll use sulfuric acid because nitrogen is one of the things that we're analyzing for. So we don't want to add nitric acid. And then for something where oxidation matters, so something like measuring ferrous iron, which is one oxidation state of iron, we want to use hydrochloric acid because it's not an oxidizing acid. There are a number of constituents where we don't use acid or we don't use a preservative. And typically for these, we will purge the sample of air to reduce oxidation and put it on ice to slow down chemical reactions. So that's typically for things like bacteria, for lab measurements of pH, alkalinity, and conductivity, and for solids measurements. So when we start considering a sampling event, we need to make sure that we have clear background information. So that includes mapping, geologic and hydrogeologic information. We want to know about stream hydrographs. So how does that stream respond to rainfall so that we can plan our sampling and not end up in a dangerous flooding situation, for example. We want to understand the land use and mining maps because typically what happens on the land is is the biggest influence on water quality. We want to understand soil maps to know what to expect, and any existing or previous water quality reports and data are going to be key information in planning a new sampling event. There are multiple sampling strategies. We're just going to cover a handful of the most common ones here. We'll talk about critical flow sampling, phase sampling, and then I'm just going to touch on long-term sampling now. Essentially, when we're setting up a long-term sampling plan, what we're going to do is we're going to look for sites that are going to be most representative of the sort of changes that we're looking for. And we're going to set up sample sites where we expect to have access in perpetuity, right? So that may be public lands, that may be sampling at bridges. That really means thinking through, trying to avoid private land that may change hands, where we may lose permission over time. And so long-term sampling ends up being something we do for monitoring long-term changes. And so there are some particular things that we think about when it comes to long-term sampling. We'll talk a little bit more about critical flow sampling now. So essentially with critical flow sampling, this is an effort to be more efficient with our resources. And so this would be a sampling strategy that we would use if we don't really care about the range of pollution, for example. We really just want to know the worst. So with critical flow sampling, what we're going to do is we're going to sample in the flow conditions that are going to cause the worst water quality. So for many, many sites, that is low flow, steady, stable conditions. Because in low flow at stable conditions, you typically have less surface water diluting your pollutant. And so oftentimes that is your most critical condition. And so if you're really just looking for what is the worst water quality, you would target that critical condition. There are some sites where the critical condition is a runoff event. So that would be something where you have exposed pollution generating material on the surface. And so that may be in an agricultural setting where maybe you have manure spreading before a storm, or that could be in like a mining setting where you have exposed acid generating material on the surface of a surface mine, for example. So alternatively, a phase sampling approach is another way of efficiently evaluating a watershed, but this looks at a bigger range of conditions. It doesn't just look at the worst of the worst. So this would be a strategy that we would undertake for actually planning projects or planning a restoration plan. We would start out with phase one. This is basin reconnaissance or holler crawling. And essentially what this is literally what it sounds like. It's hiking your creek with a pH meter, a conductivity meter, nitrate test strips, depending on what your target constituents are, and testing along your main stem and testing the mouth of each of your tributaries. That's going to then give you the information to target a more focused phase two, where you're going to do high flow and low flow water quality sampling with discharge measurements to determine which of those tributaries are contributing the most load, which of those tributaries are the most critical to the health of the stream system as a whole. And so that will then allow you to prioritize streams and plan which of those streams need some sort of restoration plan or a treatment project or something implemented that's going to improve that water quality. So then phase 3 is going to be that final phase of a phase sampling approach where, um, where a discrete pollution source is going to be sampled with the specific purpose of designing restoration or designing treatment. So this is much more focused. It will typically be more data or more frequent collection of data because it's for design purposes, design and implementation purposes. So those are just a handful of sampling strategies that we can undertake. Um, and we'll continue to think about those things as we look at specific sampling methods across, throughout this course.